Well, thank you for coming back for part 2. And yeah, it's been an exciting day. I just got the news this morning, so it's very nice to be able to be here and share it with a lot of friends. So yesterday I was telling you about evolution as a design algorithm, and I thought I would continue the story by telling you how everything I told you yesterday is slow and stupid, although I did mention that. And I'll tell you about another process that nature uses for exploring novel biological functions, and that's by recombination. Before I do that, though, I'll reintroduce the same character, which is my favorite model system. It turns out to be a very practically useful model system in addition to being a fascinating protein, and that's my friend the cytochrome P450 enzyme, which I like because it can do extremely difficult chemistry, and that's activation of unactivated CH bonds. And oxygen insertion is one of the reactions, so it's a monooxygenase. Does it at the cost of NADPH cofactors, and it does it very, very well under mild conditions. It's also extremely versatile because it can do a number of other oxidations, reactions as well. So there's a long list, and my vision is that we could actually generate a general CH activation platform just from this one great discovery that nature made. The mechanism I'm not going to go into, this isn't an enzymology seminar, but rather one on biological design, but I'll point out that it's a pretty amazing little molecular machine. It has 2 substrates. One is your hydrophobic substrate that gets oxidized, and when that binds, it enables the first electron transfer for reduction of the iron. Second substrate, which is molecular oxygen, can come in, and it undergoes a second electron transfer. So we've got 2 electrons being delivered one at a time in this catalytic cycle. And the highly reactive oxygen species is generated and then the catalytic cycle, when the product is released, to release the product, can go through the catalytic cycle. And I'll just point out that there's a little shunt reaction, which will be important for today's lecture, in which all of these finely tuned electron transfer steps, and I haven't shown the structure, this is the heme domain structure, but these finely tuned electron transfer steps can be bypassed in the presence of a peroxide. So hydrogen peroxide, for example, can deliver the oxygen for substrate oxidation as well. So you can actually undergo the catalytic cycle via this hydrogen peroxide shunt pathway, and that had been noted many, many years ago, and of course the enzyme doesn't particularly like that pathway. It usually shuts down after a couple of turnovers, but it certainly had been noted. And I'll also point out that in this wonderful molecular machine, a lot of the work is done by a prosthetic group, the porphyrin, and so a number of people think of the P450 as being all this protein gunk surrounding the porphyrin. The work is done by the heme, and the protein serves the role of providing an active site, a substrate binding site that allows substrate to come in. Obviously, that's a gross and unfair simplification, because if we realize that this finely tuned electron transfer has to take place, and also, as I showed you in the movie yesterday, the active site is opened up when the FG helices move apart, and when it closes down over the substrate, water is excluded from the active site. So it's unfair to think of this as just protein goo that provides an extra shape. That would be biomimetic catalyst designers would call it protein goo, and biochemists would appreciate it for the complex machinery that it is. And let me just say that making biomimetics from hemes has been singularly unsuccessful in providing the same kind of chemistry that the P450 can do. So it's really a wonderful object that nature has made. In fact, the heme domain responsible for catalysis, where the work goes on in substrate oxidation, is just this little green section, and there's a large part of the machinery in the reductase, and it forms a tightly interacting complex that is responsible for the catalysis. As I mentioned, a fascinating thing about the cytochrome P450 is that it comprises a very large family of enzymes that can be found in a wide range of organisms, from humans to microbes. And if you look at all their capabilities over all of evolution, at least those capabilities that exist in 2008, and probably a few years before when those things were submitted to the databases, we see that it's acquired the ability to accept a very wide range of substrates and do this versatile chemistry on such a wide range of substrates. Humans, may interest a number of you, have more than 50 of the cytochrome P450s where they're responsible for first line of defense and xenobiotic breakdown. So we'll find them in drug metabolism, but we also find them in biosynthetic roles in steroid synthesis and fatty acid prostaglandin biosynthesis and a number of other places as well. What I'm going to point out about the natural evolution of these P450s today that's important for this lecture. Yesterday I pointed out that somebody discovered one a long time ago, and over the process of Darwinian evolution, that is the creation of mutations and then their sorting out through the process of natural selection, this family of proteins that exist today was generated. And I pointed out last time that there can be a very large number of amino acids that are changing during this process, such that two P450s that exist today can differ in more than 85% of their sequence. This is out of 450 amino acids, more than 350 changes, so there's a lot of mutation that goes on. Yet, the three-dimensional structures don't change very much. And so biochemistry students learn this early on. If you look at the families of proteins that are related by evolution, we see that the sequence can vary a great deal. The function also varies, but maybe not as much. All of these are oxidative enzymes, monooxygenases, as well as the other chemistry that they can do. But what doesn't change rapidly over evolution is the fold. And so, in fact, the retention of fold is often a very good indicator of evolutionary relatedness. And this is important for today's lecture because I'm going to remind you of how evolution can be used as a forward engineering algorithm to make new proteins in the laboratory. And then we'll talk about how this sequence divergence and the retention of fold can also be used. So yesterday, I mentioned that we can make new versions in the laboratory by just essentially mimicking this Darwinian evolution, but now applying a breeding pressure. I decide who lives and who dies. I decide which ones go on to the next generation. And that's easy to do because I feel little compunction about flushing my bacteria down the drain. And I can take the gene that encodes a P450 that exists today, rapidly make many versions of that, but express those in cells, select and screen for the ones I want, and when I find improvements, I can repeat the process, thereby creating my evolved proteins with new properties. For example, I can make them more stable or more active on non-natural substrates and look for the activities that I know could exist in the natural world. I can recreate them in the laboratory. And so the good news is that this forward evolution works. I hope that I convinced you of that with yesterday's talk, but perhaps I also frightened you because on the order of one new graduate student is required for evolution on one new substrate. It's a labor-intensive process. I think I described something like the accumulation of 23 mutations to make a protein monooxygenase. And if you bothered to look at the dates on some of those publications, this has been an almost 10-year project. So if I want to explore the entire functional diversity of the P450 family, this was back last year when it was only 4,000 sequences in the database, and maybe 4,000 different substrates, and so it doesn't scale particularly well. And, in my middle age, I'm getting greedy. I would actually like to be able to make P450s that could perhaps cover the entire range of the natural activities of these enzymes. So I don't really want to follow this experiment 4,000 times, and I doubt that the 4,000th graduate student would be terribly thrilled about signing up for my group. All right, so given that, I want to talk about a second very important diversity-generating mechanism in natural evolution, and that's recombination, basically molecular sex. So if we can take the products of evolution that exist today that encode for these evolutionarily related proteins, they all share pretty much the same three-dimensional structure, but they're very, very different in sequence. And what we've learned over the years from a very large number of laboratories and biochemical experiments is that if you make chimeras of proteins, such that the crossovers between the genes, say we take one segment, the green segment from the green protein and the blue segment from the blue protein, and retain the order of the amino acids, many of these chimeric proteins are functional. You can retain the fold and the function of proteins if you choose these crossover sites carefully. So often they retain the structure, yet they can diversify the sequence very significantly. So chimeragenesis has been used to study, for example, to ask questions about what parts of the protein are conferring which function. You can use this in the laboratory, though, as a diversity-generating mechanism by making large families of these chimeric proteins all at once. And so that will be the subject of today's lecture. Let's see. In fact, it's interesting to ask this question, just how conservative would the mutations be? The idea is that you can make chimeras because the mutations that have accumulated in natural proteins, the work of natural evolution is embedded in those sequences, are compatible with the three-dimensional structures of those proteins. And therefore, if you mix and match them from different sources, they will be more conservative mutations than mutations you could make at random. So we can ask it actually in the laboratory now, because there's so many great methods available, we can actually ask this question in a high throughput fashion and say just how conservative are these mutations, because we can make, for example, if we have 13 crossovers between two genes, you can make 16,000 sequences, and if you choose the right genes, such as a beta-lactamase that confers antibiotic resistance, you can rapidly screen to identify which sequences will retain function and confer that activity. Now, just 3 or 4 years ago, when this work was done, actually 5 years ago, it was actually a little bit of work to assemble these. You could buy the oligos from your favorite supplier and assemble them combinatorially to make all 16,384 sequences, and that took about 4 weeks. And of course, the technology has changed so much that you can actually just go and have these genes synthesized at a reasonable price by a company. So it's amazing how things have changed even over 5 years. But Michelle took her oligos, synthesized the genes, made all those possible sequences, and put them back into bacteria and said, which of these encodes a functional beta-lactamase? And the first thing that she found is that, in fact, recombination is much more conservative than random mutation. Yesterday I showed you that as you accumulate mutations randomly in a protein, the fraction that retain their function drops exponentially with the number of mutations. So we see that kind of behavior. With recombination, however, we can go up into very large numbers of substitutions. And because both of the parents are functional, we know that this curve has to go down and then back up again. And so we can actually quantify using the beta-lactamase experiment where this curve lies. And so these are the actual data from Michelle's experiment to calculate the effects of recombination. And I'll just tell you, as you go out to 60 to 90 amino acid substitutions, where this exponential decline takes you down to very low numbers, we find many orders of magnitude improvement in the probability of retaining fold. This is a highly conservative operation. So if we look at the fitness landscape on average, here we're plotting the probability of folding as we accumulate random mutations. So this is no longer on a logarithmic scale, it's just the exponential decline for random mutation drops down and essentially goes to very low numbers. With chimeragenesis, you have this ridge on average in the fitness landscape. So it's a very special part of sequence space that's rich in folding and function. However, if one is actually doing the experiment in the laboratory and you're trying to accumulate as many mutations as possible, we'll talk about whether that's interesting or not, but if we want to create chimeras that are out here at 100 amino acid substitutions, the probability still drops down below what I'd want to work with. Because if you imagine the way that I do these experiments, I don't actually do them, the way the students do these experiments, we have to screen through each individual mutant one at a time and determine which ones fold, and then from those hope that we find something that still functions. So a probability that drops to 1 in 1,000 is not very much fun, because every 1,000 I'd find one that still folds, and maybe every folded one there'd be 1 in 1,000 that has an interesting function, so that's screening far too many things. So what I'd really like to do is create large numbers of proteins that have large numbers of amino acid substitutions, and I'm going to do that by recombination. But I'm going to do everything I possibly can to bring this probability up much closer to 1. And here's how I'm going to do that. I'm going to contaminate my beautiful experiment with some thought. And it's not going to be the same kind of thought as we used yesterday to argue that you really want to keep mutation levels low. It's going to be actually structure-based. So these are the kinds of fundamental questions one can ask with these experiments. When does recombination happen that's not disruptive to the three-dimensional structure? That's a prerequisite for folding and function. You want to keep the structure, but accumulate a large number of mutations. If we accumulate a large number of mutations, what kind of functional diversity do we find? And in particular, if you take evolutionarily related sequences, most of the mutations happen pretty much on the surface of the protein until you get up to very high levels of mutation. So the mutations that are happening in these sorts of experiments, where we're talking about 50% identity, let's say, those mutations are happening largely outside the active site of the enzyme. So now what you're doing is you're accumulating lots of mutations. The shuffling goes around by recombination, but they're mostly outside the active site. So it's an interesting question. What kind of functional diversity can you get from an operation like that? And finally, if you're creating thousands of proteins in the laboratory that fold and function and have hundreds of amino acid substitutions, what do you have? You have a synthetic family of proteins. So if you've got this synthetic family of proteins, and they're not just sequences in a database, they're actually things you have in your hot little hand. What can you do with that? And can we discern, for example, sequence-function relationships from those proteins? So let's go to the first question. The first question is, when is recombination not disruptive to the structure? And for this, I can go to work that was stolen out of biochemistry, actually out of genetics, and taken into computer science, genetic algorithms, and take it right back into biochemistry. And in computer science, it was recognized that if recombination is used as an operation in an optimization or a software development, then you can use recombination. Recombination gives you solutions in which things that interact with each other favorably in the solution are retained. So favorable interactions are not broken by recombination. It's effective. So there will be some partition of your object into pieces that breaks the fewest interaction. And if you have the wrong partition, you're not going to be able to build up the object from that. And so if we think of a three-dimensional protein structure, these are not domains or anything like that. This is a three-dimensional structure of the cytochrome P450. What I'm proposing is there will be some decomposition of this protein into elements that become swappable, from which you can take from different colored parents, for example, and rebuild this P450 and still retain its three-dimensional structure. One way to look at this was a nice experiment that was done fairly recently by Lemassa and Hernandez, who just took two related proteins from a thermophilic source and a mesophilic source, so this one's less stable than that one. And they recognized that these two proteins, even though they're single domain proteins, they're small, compact elements, they shared an interface. So both of the proteins shared amino acids across this interface. And when you swap these pieces of the structure, so if you swap red into the blue protein or blue into the red protein, this interface does not get disrupted. And so therefore that swap is perfectly compatible with the three-dimensional structure. And they showed not only is it compatible, you can make these chimeric proteins, but the effect of these swaps is perfectly additive. So that if you measure the delta-delta G for this mutation and the delta-delta G for that mutation is exactly the same as the other side of the thermodynamic cycle. So these effects are additive even though they're happening over many mutations. This is exactly a very nice demonstration of what we put into place several years before in the schema recombination algorithm that Chris Voigt did when he was joint student with me and Steve Mayo. And the basic idea was to identify this partitioning of the protein where we're going to recombine elements from many proteins. It can be 3, it can be 23, it doesn't really matter. But we're going going to do it in such a way that we choose the crossovers to break the fewest number of interactions upon recombination. So the question is, what are those elements that break the fewest number of interactions? And we're going to define the interactions in a very, very simple way. This is where the contamination of the experiment with some thought comes in, only the thought is very simplistic. Because yesterday I said details matter. They matter a lot, and we don't understand the details. So I'm going to describe the protein as something very simple. And that is interactions from a contact matrix and then an identity, sequence identity among the parents. So the C function is just the context. Let's take P for P domain and just look at all one another, and we'll just give it a cutoff. And so you can draw this protein like this. It is a one-dimensional chain where interactions are these connectors between the amino acids. So there's lots of long-range interactions in this protein, lots of short-range interactions, and then some of the others. And you can also draw the same thing in the form of a contact matrix, where there's a 1 if there's interaction, otherwise it's 0. Then the delta function tells you what sequence identity the parents share. And that's really critical for this, because remember there was a conserved interface that makes it so that interactions don't get broken, even though there's lots of interactions between those two pieces of the protein. So if we think of a crossover between the sequence of parent 1 and parent 2 to make this particular chimera, this interaction in the chimera, M to A, existed in one of the parents. So that interaction cannot be broken. That's an interaction that was not broken, so that'll get a delta function of 0 there. Whereas this particular contact in the three-dimensional structure, R and B, did not exist in any of the parents, and therefore is a new contact, or represents one that's broken. So when you sum these up over all possible contacts, we can score each chimera by an E-value. And then the design becomes a dual optimization, where you're trying to minimize disruption for a certain level of mutation. And this is a dual optimization problem that can actually, we can get the global optimal solution by dynamic programming, and that's what Jeffrey Endelman did a few years ago. As I say, the details matter. We don't know anything about the details, so we don't know much about what mutation level is required to obtain new function. So I'm going to try to maximize that just for fun. Does this have anything to do with protein? This is a very simple model of scoring broken interactions by $1. So it's going to cost you $1 every time you break an interaction. And I'm not going to give you any credit for making new interactions, because I don't know what those provide. And so does that tell me anything about who's likely to fold and who's not? So let's go back to the beta-lactamases, because that's such an easy system. Recombine 3 that share between 37 and 42% sequence identity, so these are not terribly identical. There's a lot of mutations there. Make all the possible sequences, which for 3 parents and 8 segments or 7 crossovers is more than 6,500 chimeras. And this is a historical slide. This, we, for technical reasons, went to a new method for constructing a library in the same year. This was a lot of work to make exactly what you can order on the internet now in synthetic genes. So we don't make this using complicated methods anymore. We just get synthetic genes to make these chimeras. So you can make all 6,561 chimeric sequences. You can sequence them very rapidly because there's only one possibility at each of the 8 blocks. I'm sorry, only 3 possibilities at each of the 8 blocks. It's either going to come from the blue parent, the red parent, or the green parent. So if you have a probe, a sequence probe for each parent, so if it's got A1 there, it's going to light up for the green probe, and so you can actually do this at 384 well plates, and you can sequence each block in high throughput that way. When you do that, we found that somebody made a boo-boo in the library construction, left out red for position number 4, but otherwise it was okay. So we know what the unselected library looks like, and we know that we can, in principle, be looking through all the possibilities for swapping fragments from each of the parents. So here's some of the ways, the new ways that you can be a beta-lactamase by recombination from this library. First of all, looking at 553 sequences, we found 111 that folded. That means that 20%, so not 1 in 1,000 now, 2 in 10 will fold and function at this very high level of mutation, up to 86 amino acid substitutions. So I don't know too many people who can accumulate 86 amino acid substitutions in hundreds and hundreds of proteins at the same time. These are all functional beta-lactamases. They have pretty high levels of mutations at the most mutated ones. There's lots of crossovers, so you can find crossovers in multiple locations that lead to functional proteins. And in this set, on average, there's 44 amino acid substitutions with respect to the closest parent. So, is there any correlation between this scoring function that I described to you, which is an incredibly simple one, this E value, this disruption value, and whether the enzyme folds and functions or not? And the way to look at this is, we know that mutation is disruptive. So, of course, things that have low levels of mutation are probably going to have a high level of function. So we expect to see active enzymes out here. But out here at high mutation levels, it's very clear that the ones that have low disruption, that make mutations that do not disrupt interactions, are the ones that tend to be active. So we know that this scoring function gives us some information for recombination. Well, if that's the case, let's go and make a whole bunch of P450s that way. So let's take cytochrome P450s that are related to one another, fairly closely related. We can take the Bacillus megatherium, one that I introduced to you the other day, yesterday, and its close relatives from Bacillus subtilis, 102A2 and 102A3. These all share about 60% sequence identity, and therefore differ at about 150 amino acids, because this is a much longer protein. These are the blocks. If we decide to cut it into 8 segments, this corresponds to 8 blocks that are color-coded here, and I'll show you those in a little more detail in a minute. And once again, this gives us 6,561 possible sequences. This corresponds structurally to sequence elements that are more complicated than secondary structures, for example. We're not just swapping in an alpha helix or beta sheet. By forcing crossover at 7 positions, and also saying you can't have very small segments of structure, the algorithm chooses the crossovers to occur at these points. And you'll see interesting things like a crossover being placed in the midst of an alpha helix. So it's not even choosing the end of the alpha helix. It's actually saying you get the least disruption from putting it in the middle. You get long meandering sequences that go through there that probably don't care where you're really cutting. You get more complex combinations of beta sheets and alpha helical regions. And this is the corresponding space filling model. And you can see there's plenty of interaction surface between each of these. We're not just looking at curls on a string and cutting where the string is. There's lots of interaction between these elements. But these are ones that have the most conserved interfaces. It is interesting, however, to look at how the domains, or what you would call them, swappable elements identified using this algorithm compare to subdomains in this protein that we know from things like molecular dynamic simulations. By looking, for example, at pieces of the protein that move in a correlated fashion, you can identify subdomains in the cytochrome P450 that have these correlated motions. They're spread out along the primary sequence such that we have orange interacting with orange over here, so recombination couldn't quite capture these boundaries. But if you look at where the schema recombination points come out to be, we have points that mimic these very, very well as you possibly can, such that block 1 and 7 together make up this subdomain. Blocks 4 and 5 make up this subdomain, and blocks 6 and 8 make up that one. So it's trying its darndest to capture those, even though there's no dynamic information put in there whatsoever. It's all structural. So there is clearly some attempt to identify structural domains. All right, what do these proteins do? That's the fun part. With the cytochrome P450s, it's easy to assay for who folds. Actually, that's actually pretty hard to do for a lot of proteins. This one we have a good folding assay because it won't bind the heme and ligate to cysteine unless it's properly folding. And in fact, it's a nice probe because you only get the difference spectrum, the carbon monoxide difference spectrum, at 450 nanometers if it is properly folded. If it's slightly misfolded, you'll get a peak at 420, and you can distinguish those. So in a 384 well plate, we can array out the clones for all the different variants and see very rapidly who folds and who doesn't with a certain particular color. So here are the results for the cytochrome P450 family. If we did a random sampling of 628, we found that nearly half encode folded proteins. This is represents about a 10,000-fold increase in the probability of folding by using the schema algorithm is our estimate. They have, on average, once you have the folded ones, 72 amino acid substitutions. So these are really significantly different. They are patentably different from one another. So that's a measure of difference. Biochemically, they're not. They may be different or not. We'll tell you about that. But they are, at a sequence level, significantly different. At 47% in 6,560 sequences, that means there's more than 3,000 folded P450s. And when I first came out with this result, the entire database of P450s was on the order of 3,000. So this had doubled the number of functional folded P450s that were known. Very wide range of mutation levels, up to 109 amino acid substitutions now. So they really are quite different from one another. And the difference between my P450s and the ones that were in the database in 2005 is that mine are all sitting in plates in my refrigerator, and I can do things with them. I don't have to synthesize them, I don't have to clone them, they're already all done. And so I can start asking interesting questions about the ones that are folded. And unlike the ones that you find in nature, I even have some not folded ones. You may think that those are not interesting, but if you've ever done computational analyses, it's really nice to have real decoy sequences. These are the ones that have gone through the same operations but did not encode a folded P450. So if you think you're good at predicting what makes a protein fold or not, I will offer you my sequences to play with. So let's ask a functional question. And this is a really interesting and simple question. I know that it interests some of you in the audience. So let's talk about how these different blocks contribute just to the stability of the protein, the conformational stability. A cytochrome P450 does not undergo reversible unfolding. So we're going to be looking at irreversible unfolding. We'll measure it, just for the purposes of this talk, by looking at the temperature at which half of it unfolds and is no longer binding carbon monoxide. So you can ramp up the temperature, see where the carbon monoxide peak is, and come up with a nice curve like this and determine the T50. Here are T50 values for about 200 of the folded chimeras. And there are a couple things to note from this. First of all, parents distribute over this range from about 44 to 55 degrees centigrade. So they're not terribly stable proteins, but they're easy to work with still. And the chimeric proteins distribute such that most of the chimeras are within the parents. So most of the children are functional, right? Half the children are functional. Of the functional children, most lie within the parents' capabilities, but there are outliers. There are some children that are less functional than the parents, and even more interesting, there's a nice tail out here where the children are better than the parents. So this recombination operation has the possibility of giving chimeric proteins that are more stable than the ones that you started with. In fact, some of the most stable P450s that have been recorded. Well, gosh, we've got 190 sequences. We've measured their stabilities. What can we do with this? How can we think about these? Why don't we start with the simplest possible model? The simplest possible model for correlating the stability of the sequence is just to assume that the effects of the different blocks are additive. So that means with 3 parents and 8 blocks, we've got 24 possible sub-elements that are being swapped among all these proteins. And if we do a linear model, that means we've got at most 24 parameters, but it happens we can use 1 as a reference, so we only have 17 possible parameters to fit in here, A0 and then 16 of these elements, where each fragment, Xij, is contributing Aij in terms of stability. That means you can take a chimeric protein and just sum up the contribution of each fragment if stability is additive in this system. So why not? Let's give it a try. We looked at the data for that and were stunned to see a really good correlation for this simple model. So if you predict the T50 by fitting all of the 204 points to the measured 50, you you do the model, you find a fit, and then you predict then the T50 from that fit, you find a really nice correlation. In fact, the correlation is so good that when we went and looked at the worst outliers and went back and sequenced them, they had sequencing errors. So all the other ones were perfect. All the blue ones, the sequences were perfect, but we found point mutations in these and sequencing errors from the probe hybridization, which has a 3% error rate in the worst outliers. So it was a pretty good fit. All right. If it's a good model, then you can predict things that you did not know before. So what you'd like to do now, if you have a good model for linear fit for what the contributions of the different sequence elements are, you should be able to predict the stabilities of new proteins, of new chimers that had not previously tested. So in fact, we went then and found 20 new sequences, just pulled them out of the soup and constructed a few, including the predicted most stable chimera, and found an equally good fit for those predicted stabilities. In fact, the most stable P450 has a stability on 65 degrees centigrade. Wow, that's pretty remarkable. What that means then is if your model is good, we should be able to take a small sampling of all 6,561, take a sampling experimentally, measure those, and predict all of the stabilities of all the proteins in that set. And the most interesting ones, of course, are the several hundred here that are more stable than the parent, the most stable parent. So if we're so smart, we should be able to measure this, predict the most stable ones, synthesize those, which we did, and construct them and test their stabilities. So we took the 44 that were predicted to be the most stable, 44 of the most stable from the 300 that were predicted more stable than parent A1. So these are their sequences. You can see that there's a consensus here that parent number 2 should be at position 1, and then there's a little less tight in other positions. And when we synthesized them, every single one of the 44 was more stable than the most stable parent. So this was just published in September. But we have now 44 highly thermostabilized cytochrome P450s, whose stabilities range from the T1, which is about 55, 56, to 65 degrees centigrade. So that just shows that the schema algorithm was able to capture the interfaces, capture the cut points that allow these pieces to be swapped. And not only that, then allow these pieces to contribute in an additive fashion. This is just a demonstration of the sequence diversity that's available. We can put this on a ternary diagram where the sequence of A1, A2, and A3 are the vertices of this diagram, and then the sequences of the mutants or the chimeras lie in between. And this is just to show that the sequences are different from one another in a significant way as well. They're not 44 of the same protein or 625 of the same protein. These are all significantly different from the parents and from each other. So who cares about stability? Stability is great, but it's only interesting in the context of what a protein does. Right? Having a stable rock is not as interesting. So what do these do in terms of their catalytic activities? And let me tell you just a little bit about that in the last few minutes. As I mentioned yesterday, BM3, in fact, all of the parents for this experiment have very nice properties in terms of their biotechnological properties. They're well characterized, they express well in E. coli, all the work is done in a single polypeptide, but they have this fatty acid hydroxylase activity that is of marginal interest to me. It's very fast, but it doesn't do anything particularly interesting. And so what I'd like to do is be able to screen them to find out whether these new recombined proteins that have many amino acid substitutions have acquired new catalytic activities. And for this, I'm going to make use of this peroxide shunt. Because remember, I've only done this in the heme domain of the P450. I don't have reductases attached to these. And so instead of using the monooxygenase activity, I'm going to feed them hydrogen peroxide and drive the reaction through the peroxide shunt. And there's a particular mutation that had been reported to increase that activity many-fold, and that mutation was made in all of the parents. So they all should, if they are active, have this peroxygenase activity. So how do you find out what's the range of activities in these enzymes? Well, you have to go and measure it. And so like a 64 question type of game, they're not going to tell you what they do unless you have a way of asking the right questions. So you might have your cure to cancer there in that library or the energy crisis, but if you haven't asked it, it's not going to come out and tell you that. So that just means you have to knuckle under and develop high throughput screens that give you a nice colorimetric output or fluorometric output for these activities. And we have those. That's exactly what we've been doing for the last 10 years with the random mutagenesis. So we have activities on fatty acid-like substrates. Aromatic hydroxylation is easy to measure with 1920s German analytical chemistry. We've got our demethylation assays. When you make formaldehyde, then you get a nice purple color. And you combine all this, we've got alkene epoxidation assays. So we just collected all the assays we developed over the years and started asking each of these chimeric proteins, you've got the heme in there, what can you catalyze your reaction on? And I'll summarize quite a bit of work just by telling you most of the ones that fold also function. So that little bit about the protein goo around the heme domain is valid if you have a heme, if you've got a heme properly inserted into the protein, it will be active on something. Reverse approximation. What it's active on, you've got to find it out, but it will be active on something, which is a nice thing for the cytochrome P450s. So you have to knuckle under and do some real work. So we took 14 of the best expressed chimeras, as well as the parents 1 and 2. The heme domain of parent 3 is not functional all by itself. It requires its reductase domain to be functional on any of these substrates. But we can measure the activities on the substrates. And I'm not going to go through this data because it's mind-numbingly boring to look at these kinds of plots, but I will tell you the conclusion that's important for this lecture, and that is for every single one of the substrates that we studied, and none of these look much like fatty acids, there's at least one of these chimeras in the 14 that's more active than either of the parents. So just by checking 14 chimeras, we found something for every substrate that's more active than 1 and 2. Furthermore, you can find substrates that on which the parents don't have any activity, so 2, 3, 7, there's very little activity or no measurable activity in the parents, yet there are chimeras, and sometimes multiple cases, where you can find activity. So you can find new activities from these chimeric enzymes. For example, if I introduce you to some of the chimeras, 21113312, which looks like this, is active on zoxazolamine. That's a human drug, and it's a substrate for the human P450 CYP1A2. CYP1A2 takes about everything. And this chimera will make the identical human metabolite made by the human enzyme. They make the same product. And if you just plot where the amino acid substitutions are, this one is 98 different from its closest parent, which is green. And the 98 amino acid substitutions, as I told you, are lying almost all on the surface of protein. There's nothing near the active site. They're all just here on the surface of protein, yet it confers this new activity. 21313313, which looks like this guy, demethylates permethylated sugar. So I introduced that reaction yesterday, and this is one of the culprits that does it. It's differing from its closest parent at 85 amino acids, once again, primarily on the surface of protein all over here. And this will take the 1 position and do a selective demethylation on that. And finally, here's one more example. 21312332, which looks like this one, is different from its parent at 84 amino acid substitutions. And yet this will take verapamil and make the authentic human, a single authentic human metabolite, so it will demethylate, will dealkylate at the end position here to create these two products. And that's an identical reaction made by rat liver microsomes. So these are functional enzymes, and they're actually pretty good. So these are also part of the ones that have gone into the Codexis CYP set. So let's finish with one last little functional thing. And that is, remember, we're only looking at the heme domain. I've made these recombinants of the heme domain, yet we know in the whole enzyme complex it's interacting with the reductase. So if I would like to reconstitute the monooxygenase, how can I do that? Which of the 3 possible reductases should I use, or do I have to recombine the reductases? So we went and made all the parents and 14 of the active chimeras, the ones that we studied before, and reconstituted them with all 3 reductase domains and asked which of these will be functional. And what's interesting is that every single one of the fusions work as holoenzymes. And why is that? I bet you can tell me by now. The interfaces are conserved. All of the key interactions between the heme domain and the reductase domain are conserved among the parents. So are almost completely conserved. So the blue ones are conserved in all 3 parents, and the red ones are conserved in 2 out of the 3 parents. So these key interactions across hydrogen bonds, metal binding interaction, water interaction, are conserved among all 3 parents, so that when you swap them, it doesn't really care. So here's something, these reductases differ at 50% of their sequence, yet they retain this ability to interact across the domain. Those were retained during evolution and therefore get retained when you do the swapping. All right, so let me summarize some of the points I've tried to make. The first question was, when is recombination not disruptive to the structure? Well, that happens when blocks are defined by these maximally conserved interfaces. And the schema scoring function allows you to identify those computationally in a very rapid way. And so that when you recombine these blocks from different parents, you can make proteins that are highly mutated, yet still fold and function. So we can make 4,000 or 3,000 new cytochrome P450s in a single experiment. What functional diversity is accessible? Well, I've just shown you that in a very superficial way, but I hope that I've shown you that there is functional diversity there. They exhibit features of stability that are outside of the range of the parents, and catalytic activities that are both better than the parents and allow you to have catalytic activity on substrates that are not accepted by the parents. And notably, these bacterial P450s can acquire activities of the human enzymes. And finally, the last question was, can we discern sequence function relationships with this synthetic protein family made in the laboratory? And I hope I've shown you that. I did not discuss this here, but it's been published. We can find actually specificity determining features in one of the parents that were not known before. What I did show you was that the stability contributions are additive in this system, and therefore we can predict the stabilities of unknown sequences with a high level of accuracy using, and this is what we're proving now, a minimum of very small number of sequences. This means that even in the absence of a high throughput screen, if you can look at even 30 sequences, then you can make large sets of proteins and accurately predict highly stable sequences from that. And I didn't have a chance to tell you about sequence analysis, but I know that some people here are interested in that. These consensus energies that you can calculate from sequence analysis can also be calculated in these non-natural protein families, and this allows you to predict stabilities just from folding data alone. You don't even have to measure the stabilities. You can accurately predict stabilities just from the sequences of folded proteins, and if people are interested in that, they can go to the paper that was published in September. So with that, I want to quit, and I think time is up. It's hard to tell the timing, but I'll thank the people who did all this work, and I'll thank you for your attention. The only way to get mutations by recombination is if the mutations exist in the parent. And so, if you take proteins that are 50% identical, that's a very high level of identity. Very rarely will there be mutations in the active site. So since they don't exist in the parents, they're not going to exist in the chimeras.
So these are very highly homologous active sites.
They're homologous and they're high levels of identity. So 50%. Now that's high from an evolutionary point of view, but it's very low from a protein engineer's point of view, because you still get very high levels of mutation, but you don't get them in the active site. So this experiment shows that mutations outside the active site can confer these new substrate specificities. Whether that's a special feature of P450s, I don't know yet.
I actually have a related question. And that is, I don't know if it's possible in the P450 case, but in at least some cases, presumably one could find structurally homologous proteins that are functionally different. And so then one wonders, well, what would happen? Is that a way to get a lot more functional diversity out of, if one wants it, out of the progeny of this sort of a process?
Yeah, that's an interesting. So as you go to more sequence divergence, right, you can actually find things that have now diverged in function, or maybe converged, and so they're non-homologous. Right now, we're still limited to about 30%. 30% identity is the lowest that we've gone. So even at 30% identity, we know that they've diverged by evolution. There are a couple of bold people in the group who'd like to test what you're talking about, real non-homologous. But as you know, the mutation level still matters. And as you go to 20 and 15% identity, the structural divergence has now gotten so large. You know, it goes up exponentially with sequence divergence, so that there's a lot of disruption.
But I wonder if there's yet another variation, and that is if you take these fragments, these domains that are identified by the schema approach, and then just search for those, and then just focus then on the domain that's important, or the subset of domains that are important for the active site, and leave the rest the same.
Yes, so that's another way, but then you don't get very many proteins that way. So if you just choose one fragment, yes, you can swap in a structurally homologous fragment from another protein, but you just make one new protein. I would make 4,000. So I'd have to find 4,000 structurally homologous fragments to put in. So I don't get the combinatorial diversity.
No, but I guess that's what I'm asking, but you can find lots of different, if you just focus on the fragments, whether you could find lots of them scattered in different.
You certainly can. You certainly can. And, you know, now that we have gene synthesis, you could just as easily make that library as synthesizing all these genes. So it becomes more practical to do that. We wouldn't even have thought of doing that a few years ago because you'd have to make each one of those one by one. Whereas I could just make it all 6,000 at once with the methods we were using before. But yes, we should go back and look at that. The mutation level is not going to be as high. Right? We get hundreds of mutations because we're swapping lots of fragments at once. If you're just swapping in one fragment, then you'll have a lower mutation level. But I think that's interesting.
Is schema available for public use?
Yes, it is. All the calculations, all the calculations are available on my website. What is more difficult to do is the RASP algorithm that does the calculation of the optimal crossovers, but anybody who's ever asked us to help with that, we're happy to do. Actually, I think there may be a RASP Python code on the website now.
I have a question. The substrates that you chose to look at, so you chose a subset of compounds. It seems like, really anything's possible. Why those compounds?
The ones, the 14. Oh, yeah. You know why? Because it's really hard to measure whether a steroid has been hydroxylated. Whereas if you hydroxylate an aromatic, you can use these colorimetric reagents. If someone pays me enough, I'll go and look at a steroid, but that's why Codexis has these proteins, because I really don't want to do that. That takes LC and GC, and it's not fun.
You had the statistic where 80% of the actively folded protein or folded proteins were active.
Yes.
So, and then you showed the novel functions of 14 of these.
Mm-hmm.
So did you measure the closest parent functions as well? And was there any drop?
Oh, yeah. So the parent functions, most of the substrates that I showed you, the parents have very low function on. So we know the parent functions on those substrates. And usually we choose things that parents are not active towards.
Right, but then do they still confer the parental activity for the other substrate?
Oh, for the other substrate. We haven't looked at the fatty acid activity for the chimeras. We did that for the PMO that I described yesterday, but we haven't done it for the chimeras.
I thought the thermostability stuff was really interesting, that how much of an increase you can get in the temperature stability. Have you thought about doing this with thermophilic start points or enzymes, and you think the same increase in temperature stability would be possible?
So as you go to higher and higher stabilities, there are going to be fewer possible sequences that are more stable than those. It certainly would be interesting to find those few possible sequences, because I think the temperature limits to stability of proteins is dictated not by the physical chemistry, but by other biological requirements. So I think it is possible to make even more stable proteins than we have now. I haven't yet done that, though. It's not going to be as easy to stabilize a thermophilic protein as it is to stabilize a pure protein.
Sort of along those lines with increasing stabilization of the protein, if you run through multiple rounds, do you expect that you'll get another increase? Or because the blocks that you're using are the same blocks that you used before, so presumably that would go.
Yeah, you wouldn't do another round of recombination like this. What you really want to do is take those stable ones and find some other way to increase their stability through point mutation, for example. Or making more crossovers. Another would be to say, instead of using 7 crossovers, make 23 crossovers. But then the combinatorial explosion goes up, and finding the linear model, the linear fit, would take a lot more experimentation. So I think the better thing to do would be, you've got a nice starting point of 44 new sequences, find one that has an interesting activity and stabilize that.
Going back to the where you had basically a bunch of mutations out in the secondary shell that were affecting the substrate specificity. Have you determined for any one of those whether or not it's a single domain swap that confers that the initial activity, or is it the combination of those 75 or 80 mutations that give the just that right fit to confer that?
Right. So basically, you're asking what's the minimal number of mutations required? That I have absolutely
Okay, so you
no
haven't
idea.
gone through and deconstructed
No,
the
and we don't have enough data, say, to do a machine learning. You'd like to find what's the structure-activity relationship, and you actually need a lot of data for that.
Yeah, I mean, because I think it's extremely powerful looking at this it's easy to go in and mess with the active side, change a couple of mutations, but once you get out to that secondary shell, it seems extremely powerful.
Yeah.